Well, hello pointers, and welcome to today's lesson on mitosis and cytokinesis, the process of dividing up the nucleus and then the cytoplasm. Like last time, there will be a short quiz and some differentiated instruction based upon how you do on that quiz. So feel free to take notes, make drawings, and things of that nature that's going to help you out in the long run. Starting off, first stage, first thing of mitosis that you should know is that it's a lot like picking teams in gym class. If you look at the picture on the bottom here, you notice kids are lined up and they're sorting into different teams. Mitosis is the same way, except it's with the nucleus and its contents, particularly the pieces of DNA that are selected to go to different cells. And the idea is that you're going to equally divide that nucleus up into each of the new cells. And the idea is that way the cells will be identical and equal. In gym class, when we divide teams up, we want to make sure those teams are equal so that one team doesn't dominate the other and that they each can do basically the same thing, and that makes for a fair and competitive game. So that's the purpose of mitosis. It's a kind of a drawn out process. It's not a simple matter of simply ripping something in two to divide it. You have to go through the stages. And we'll talk about those in a bit. So one of the things about mitosis before it can happen is we have to make a copy of the DNA. So before the cell divides, remember S phase from the cell cycle, these chromosomes go from a single strand of DNA to two strands, and they are hooked together so that the two strands don't get mixed up with all the other chromosomes um, in the cell. So right before the cell divides, we see this X-shaped structure called the chromosome, and that is the two pieces of DNA that have been copied. So an X-shaped chromosome is actually made of two different pieces. The daughter cells at the end are going to get one of those two chromatids, pieces of the chromosome, and so that the end product, the two daughter cells, are identical to each other, and they're identical to the original cell. Some definitions we have to get down here dealing with DNA in its basic state in the cell. When it's in the nucleus, DNA is like a long string. And if there's more than one piece of DNA in a nucleus, we have several different strings. So if you look at this picture here where it says chromatin, you can see there's different colors of these strings, and they're all balled up together inside of the nucleus. If you were to try and separate this into two different equal piles, it would be a nightmare. It looks like it would all get tangled up, and it'd be hard to tell which color you've got and which ones you don't, and where one starts and where one ends. Chromosomes have a way of getting around that. The DNA coils itself up, supercoils itself up into this X-shaped structure that we call the chromosome. And this is a tightly supercoiled piece of DNA and its replicated identical partner. They're joined together in the center here by this structure called the centromere. Centromere is where the two chromatids connect with each other. And the individual piece, the one piece of DNA, is called a chromatid. So each chromosome consists of two chromatids joined together at a centromere. All right, so it's 3 or 4 different ways of talking about the same stuff. It's still DNA, just depending on how we are defining it. All right, so a good way to kind of wrap this all and put these pieces together. Before the nucleus can divide, before it divides, the DNA is all loose and stringy, called chromatin. That chromatin must condense and form this X-shaped structure called a chromosome. And looking here, you can see how it coils itself around these histone proteins, and then the coils coil around themselves, and then the coils of the coils coil around themselves, and it gets more and more tightly coiled until it shows up in this structure. When it gets this X shape, we call that the chromosome. Chromosome consists of two chromatids. Each chromatid is an individual piece of DNA that's identical to each other, and they're joined at the centromere. In the end, each daughter cell is going to get one of those pieces of DNA. So a little bit of a difficult concept here, how to put the pieces of chromosome, chromatid, DNA together, but that's as good a definition as I can come up with. All right, so the process of mitosis is a lot like picking teams in gym class, again, to try and keep things equal. And like in gym class, there are phases that we do that. First, the captains come out front, or first you line up on the end line, or somebody flips a coin. There's all these phases. Well, mitosis is the same way. We've got phases named prophase, metaphase, anaphase, and telophase, where certain things actually take place. And we're going to talk about these in more detail as we go forward here. Let's start right off with the first part here. It's called interphase. Now, this is actually not part of mitosis. This is what happens between the cell divisions in the cell cycle. So this is the G, the S, and the G2 portions of the cell cycle that we learned about in our prior lesson. So the things that happen during this interphase between the divisions, we have their normal cell growth and the cell activity. We have the copying of the DNA. We have the final cell growth and the final checklist, and now the cell is ready to divide. All right. So once that happens, we go into mitosis. Mitosis starts with a phase called prophase. And again, you may want to write these down and make some drawings of these so it'll help you identify what's going on. But if you look here in this drawing, you can see that our nuclear membrane is starting to dissolve. The dotted lines are showing that that membrane is going away, and our chromosomes have now condensed into this X-shaped structure. They're no longer a big tangled ball. They are separated from each other, and they are condensed that way. And once the nucleus goes away, these chromosomes are free to kind of float around the cell for a little while and separate. I liken this to in Phy Ed class, in gym class, people milling around at the start. Feel free to walk around the gym wherever they want to, not in any order, just kind of free to move about and talk to whoever they want to. Eventually, though, the teacher is going to blow the whistle and you're going to have to line up to pick teams. And that is a lot like phase 2, which is called metaphase. Chromosomes are going to line up in the center of the cell in the place called the equator, and these strings called spindle fibers are going to come and attach to them, which will later be used as part of the division process. So the spindle fibers are produced by the centromeres, if you remember that from the chapter on cells, that are at opposite ends here. And so these centromeres are going to come and attach to these chromosomes. This is a lot like when students are going to line up at center court and then be selected into their two teams by the captains. And this will ensure when they line up that nobody's missed and that one team doesn't get all the good players and the other team gets all the bad players, and that they should be divided up rather equally. Once they've been lined up, now we have to divide them. So once the captains make their selection, they divide, and one team goes to one side of the gym, the other team goes to the other side of the gym. This is called anaphase in the cell, and what happens is these spindle fibers shorten, and when they do, they pull on their individual chromatid and pull them to the opposite ends of the cell. So what we're going to end up with is this set of chromatids going to the top and this set of chromatids going to the bottom, and they've been separated into their two teams. If you think of like animation, animated things are moving, and anaphase is the moving phase of mitosis. Once they've gotten to the other end of the cell, we see they now have kind of bunched up again. They've uncoiled, unsupercoiled, and gotten long and stringy again, and they've kind of intertangled just like at the beginning of the interphase. And what's happening here, it likened it to the teams huddling up to discuss their strategy before the game starts. And around their huddle, we're going to build one of these new wrappers, a new nuclear membrane. And so that starts to appear and kind of keeps these sets of chromosomes, chromatids, separated from each other so our two teams can't re-mingle with one another. At the end of telophase, we're going to have two new completely identical nuclei, and we've got them both in one cell. Now, this isn't going to work in the long run. The cell can only have one nucleus, so therefore we have to divide the cell, and this phase is called cytokinesis. In cytokinesis, we're going to pinch the cytoplasm together in the middle of the cell, and we're going to eventually pinch this apart, and these are going to form two new daughter cells. Again, that's the process of dividing up the cytoplasm and the materials. It's called cytokinesis. At the end of this, we're going to form two new daughter cells, and they will look like this. If we look at these two things, compare them to each other, we can see they are basically identical. They have the same four colors in their nucleus: red, green, blue, and yellow. They're the same size. They have all the same instructions, and in all ways they're basically the same. These cells are then going to go back into the interphase, the G1 of interphase, and start the process over again. If we look at these cells, we can see they have those four colors, and if we go all the way back to the original cell in interphase, we can see it also had those same four colors, the only difference being that it was larger in size than our end product daughter cells. And if we did that correctly, this is what we should have. They're identical to their parent and identical to each other. And that's the process of mitosis, and it happens over and over again for cells that are needed for replacing dead or dying cells. It's used for asexual reproduction, and it's a process that has been happening ever since your life began. So again, there'll be a quiz on this tomorrow. If you need to go back and review this lesson, please feel free, and I will see you in class tomorrow. Good luck.